Okay, good morning. Thanks to everybody for joining. This is Theo Dreher from the Department of Microbiology at Oregon State University in Corvallis. My background is as a molecular biologist, mostly working over a career of 20 years or so in viruses. Over the last years, as department chair here, I've become aware of environmental issues involving cyanobacteria, particularly in the Klamath Basin in Northern California, and actually throughout Oregon. We are seeing an increasing frequency of harmful algal blooms caused by cyanobacteria in freshwater systems, predominantly in reservoirs and lakes. So these are the so-called cyanoHABs, HAB being the shorthand for harmful algal blooms. So I'd like to tell you today about the work that we've done, certainly building on previous work from a number of labs in other parts of the country and around the world, with the aim of moving towards genetic testing, that is DNA-based testing for understanding the biology of these algal blooms and for monitoring, for routine monitoring. It's something that it's a field that is at the stage where the theoretical knowledge and the practical capabilities are really there, but we do have to figure out the best way to apply the knowledge and the techniques that we have to the field. And so it's something that we're not going to be seeing in routine monitoring for a few years, but I think it's important to consider this and to start moving in that direction. So this shows the current status that you would be familiar with if you're a lake manager or work for an environmental monitoring agency. With regard to the toxic cyanobacterial blooms, there are a number of genera that are associated with toxins. Microcystis is a very common one, Planktothrix, Anabina. These produce the microcystins and the anatoxins. And so what we see here is that routine monitoring involves phytoplankton analyses, as we see over here at the left, which is a microscopic-based, that is direct microscope observation, based identification of the planktonic and filamentous cyanobacteria that are present. On the right, we see an example of a toxin analysis, a result of a toxin analysis. These are becoming much more frequent. The prices come down. The accessibility has increased just over the last year or two. So that we're seeing more of these ELISA, enzyme-linked immunosorbent assay-based toxin analyses. At the top right-hand corner, then, are the action criteria that are used by the Oregon Department of Human Services. These are based on World Health Organization recommendations. In California and Washington, rather similar action thresholds are used. For cyanobacteria in general, the action threshold, this is to direct posting of a lake as containing a potentially harmful toxic algal bloom that should lead to restricted contact by people and pets with the water. For cyanobacteria in general, the action level is 100,000 cells per mil. For Microcystis or Planktothrix or also Anabina, for known potentially toxin-producing genera, the action level is 40,000 per mil. This requires the identification of these organisms. The alternative action criterion is based on the toxin analysis itself. So in the case of microcystin, this is 8 parts per billion in terms of with regard to recreational exposure, or 1 part per billion in drinking water. So the genetic capabilities don't directly speak to relevance or represent an alternative way of measuring the toxin. The toxin measurement is really the end point that is important for public health decisions. But what I'll be telling you about in this webinar is what I think is really a more accurate, a better, a faster, probably in the long long-term, certainly cheaper way to identify the cyanobacteria that are present. So for any action criteria that are based on identification and cell counts, I think there's a lot of potential for using genetic methods. Then, in terms of understanding the blooms that are occurring, digging deeper into the population structure and understanding what is happening during a season, what happens in subsequent seasons, comparing blooms in different parts of either disc geography or close by. This is where DNA-based analyses is going to deliver a lot of possibilities. So let's go through some of the difficulties that we currently have with morphological identification of the cyanobacterial blooms. That is this microscope-based identification. What we see here in the middle is a phylogenetic tree. That's a tree that plots the relationships between identified organisms based on their DNA sequence. These are sequences drawn from the GeneBank DNA database, and I've highlighted Microcystis in the sort of orangey color. And what you see is that there are different species listed. Microcystis aeruginosa, next, for instance, in the middle to Microcystis wesenbergii. Whereas there are other entries of aeruginosa down near the bottom of the diagram. Further, you can see that Microcystis is scattered throughout this whole figure with different genera, Pseudoanabina, Anabina, Cylindrospermopsis, Oscillatoria placed in between. These names are listed indicate the closeness of relationship. So what diagram is telling you that Microcystis, all these organisms that have been called Microcystis, based on morphological identification, they don't have DNA that is very closely related. This is a problem. So somewhere along the line, there's been a misidentification. The problem, or a further problem, is that that a given Microcystis strain, a culture, either in the lab or this has been seen in nature, can assume different growth habits, and we see some different colony morphologies here in this slide. Actually, they can vary quite widely, and of course, colony morphology is something that one would tend to want to use for morphological identification because the single cells are very small and don't present very much identifiable, many identifiable characteristics for identification. So, in Japan, by Otsuka et al., for instance, that's mentioned here, has concluded that the a range of Microcystis species listed here, aeruginosa, ichthyoblabe, and so on, actually represent, or their contention is that these represent different morphotypes, colony morphotypes of the same organism, and that really we should be calling all of the Microcystis at this point Microcystis aeruginosa, unless through genetic classification means we can start to divide species based on sequence relatedness. So the idea is that it's hard based on microscopic observation to be certain what is Microcystis, even distinct from some other genera, but it's also tempting to call Microcystis that has different colony types a different species to another colony type, but there is no genetic reason to do that. In this slide, we see that subsequent to the very common treatment, Lugol's iodine, that is that samples are subjected to before they're delivered to a laboratory for the microscopic identification, there can be quite large colony morphology changes. So the fresh sample on the left and the Lugol's treated sample on the right, which is where the colony is largely dispersed. And if there's a good deal of agitation during shipping, these cells can just become entirely planktonic. And then it's very, very hard to identify them as even Microcystis. So to identify cells down to the genus level. For the fermentous genera, such as Anabina, which are like necklaces on a chain, the Lugol's treatment can frequently lead to disaggregation of the filaments, so that again you have single cells. So these various issues that really exist with the current morphological identification. In the background, the last decade or more, there have been very large numbers of genetic studies in the literature, so that there's really a lot known about a certain number, a couple of genes that can be usually used to identify. There's a reasonable database. We'll get back to that point later because it's really a little bit small at the moment, and that's one of the current limitations. But there's a lot of information in the academic literature. As I alluded to at the very beginning, is that we need to work at making this more accessible and more applied to more routine sort of monitoring. But we certainly have a very good basis in a large number of academic studies. So my view is that in the future, monitoring and the data that goes into monitoring into decisions, action decisions about whether a lake should be posted or not, could look and probably should look more like this. So certainly toxin analysis over on the right. This is going in very much the right direction now, and this perhaps would be relatively unchanged. But whether or not there would be the phytoplankton analyses that I now have on the bottom left, the morphological identification, whether or not these remain relevant and useful is a question. But certainly they should be there should side by side be the accumulation of genetic information that would deliver either the sequence shown in the top left white box there, or that sequence can be that information can be displayed in the form of a phylogenetic tree, as is shown underneath it, where intention is to show the relationships between a given isolate taken from a lake bloom in a lake with other isolates that have been previously studied. Now, one might also, and so with regard to identification, this should really, I would say, move in the direction of being genetically based rather than morphologically based. On the top-hand corner, you'll see now that it looks very much like the previous slide, except in cells I have genomes listed. So we'll see some examples of how genetic technology can be used to assess the number of genomes. Most is one genome per cell, unless a cell is actively dividing. So rather than measure the number of cells, which can be difficult when you have a very concentrated colony, as you saw in some of the earlier pictures of Microcystis colonies, one would measure the DNA content, the specific cyanobacterial or specific Microcystis or Planktothrix and so on DNA content, and thereby estimate the number of genomes. So it'd be in the future that for monitoring we would be measuring genomes as a substitute for the current cell counting. If there are any questions, do go ahead and use the *6 to unmute your phone and sing out, or we can go through the chat. I can see the chat box if you want to type in a question. So one thing that is needed before genetic information can be most usefully needed is a much larger database of the gene sequences of the cyanobacteria that are forming harmful algal blooms, say in the Pacific Northwest, in California, in any geographic area that we're concerned about. Because at the moment, although, as I mentioned, there's a lot of academic literature that has led to a large amount of information, most of this has been on a fairly restricted number of isolates. So we really don't yet have a big enough database to be able to make the sort of comparisons we want. So for instance, in working in Oregon, and I think this should be happening in various parts of the world, is to develop a local cyanobacterial database where we have samples, say, here from Willow Creek Reservoir, record what the origin of the sample. We perhaps have a microscopic photograph, because certainly looking at the cyanobacteria conveys a lot of information to people who are out there in the field. We have the morphological ID, that is the species, genus that can best deduce from microscopic observation. But then also in the blue part of the panel below, the ID based on DNA analysis. It can be based on a number of genes. There are 3 different genes listed. We'll talk about that a bit later. And on the right, you see the actual gene sequence, or part of it, for one of those, again, a phylogenetic tree. And so if this would be accessible, that DNA sequence can be retrieved by a person, it can be queried against the database, one can pretty quickly find out where the closest relatives of this strain isolated from or present in Willow Creek Reservoir have been found, and whether in fact there are close relatives within most of the water bees in eastern Oregon, where this is from, or if there's actually a rather larger sort of diversity. So we do need to work at building up this database so that there are more comparisons to query for incoming samples to be queried against. So I've mentioned some of the advantages that I see for a DNA-based monitoring and just in general research venture. In particular, I'm thinking of the lake and water managers. You know, what can this technology do for decisions on the ground about posting, about maybe long-term remediation of blooms that occur every summer? So I alluded to more accurate bloom identification. So there's the possibility, or there's certainly a DNA-based technology can identify species and even strains, and I'll give you some examples of that with some of the data we've generated, very accurately and reproducibly, so that the same person in a different part of the world would come up with the same result. And that really isn't true for morphological microscopic-based identification. There's a lot of subjectivity in that field. Quantitation. So at the same time as identifying the type of DNA sequence that's there, we can also measure how much. So that's important. This sort of information allows high-resolution comparison between lakes that I've mentioned a couple of times. One would be interested in how, for instance, blooms might be spreading. Blooms are appearing with increasing frequency and in areas that previously haven't had reported blooms. We know where those cyanobacterial strains are coming from, and if they're identical to if a new bloom has identical strains, say, from the next watershed across, then it seems likely that it may have been seeded from there. And so we should be able to figure out something about the epidemiology, we can call it, or where strains are coming from. Detection of toxin genes. So one of the genes that I'll be talking about in our research with Microcystis is one of the genes that support biosynthesis of the toxin. So as a surrogate for direct measurement of the toxin by for instance, the ELISA assays that are becoming quite popular, one can measure and identify the toxin genes. Now, a cell that has the toxin genes isn't necessarily expressing, that is, making toxin all the time. So there are real advantages to measuring the toxin. But, for instance, I mentioned here early warning detection. So the DNA-based assays can be very sensitive, and it might be useful in particular instances to measure the in a very sensitive part of the range, the appearance of toxin genes in blooms as they're developing as an early warning sort of indication that some time down the road, in a few days or a few weeks or a month, there might now be measurable toxin. But DNA-based analysis would be more sensitive than toxin analysis in these early stages of blooms. To have a more accurate understanding of bloom populations, that is, I'll give you a case history example from our studies that indicates the sort of understanding you can get of bacterial populations that is needed if we're understanding how and why they develop, rather than just globally because there are too many nutrients. How one might be able to manage this to change the population, either to make it less toxic or to really have it, again, brought into control. At the bottom I have high throughput detection. The DNA sort of base techniques can be designed, can be implemented in a high throughput sort of mode, such that after sampling and DNA extraction, then you can, for instance, use a 96-well plate. You could ultimately use robotics to push through a lot of samples. So that means that per sample in that format, the cost is cheaper. The increased speed. One can think of sampling more sites at different depths and so on. And because sampling techniques are always a bit of an issue as to how representative a particular sample is of the situation in a lake, there are real advantages here. So how do we get these samples? There's certainly a lot of technology that perhaps many listeners are unfamiliar with. I notice with students in lectures that often this can be a little bit daunting. So let's just give a brief introduction of roughly how the samples are handled in very simplistic sort of terms. Sample collection is the same as it usually is. You go find a bloom. You can either sample integrated depth. You can take the scum, which is perhaps that's floating on the surface, which is perhaps from a public health point of view pretty critical because kids might put that in their mouth and so on. And then currently we're handling samples that have been then stored cold, shipped on ice, and not preserved. It should be possible to actually preserve in the usual sort of way with Lugol for some services with glutaraldehyde. Then you wouldn't need the cold preservation and shipping, and that would presumably be a cheaper option. So bottom line is that sample collection and handling and shipping isn't very different from what's normally done. In lab then, we filter the colonies and collect that DNA and archive it in the freezer. And that can be archived then for a number of years, or it can be immediately studied. Then a number of DNA-based assays are applied. And this slide here represents the polymerase reaction, or PCR, which you've probably heard. The important issue about PCR is that we target it at the genome, that is at the DNA, at the chromosomes that are present inside the cells, represented in that first orange double line at the very top. The neat thing about PCR is that it utilizes small synthetic molecules, small synthetic DNA molecules that we refer to as primers, that are indicated by the small green and blue boxes that are now opposite or adjacent to those two single orange lines, which are now separated. So DNA, as you know, is a double helix. That double helix is shown at the top. By melting in a machine, the helix is broken so that we have the two separate strands. And that allows the green or the blue primer to bind to an appropriate site. The appropriate site is important. That can be designed because we need to know the sequence of the DNA, and we know a lot of sequences of cyanobacteria. the neat thing is then that one can design the primer specifically to target gene A or B or X or a particular part of it. So we have entire experimental control over what part of the DNA is targeted and then amplified, because the bottom two-thirds of this slide shows cycles in which each time there is a blue or a green box bent to a single orange DNA thread, the next line shows that the polymerase has now made that second strand of DNA, and we have a double helix again that incorporated those coloured primers. So we go from having a single double helix to two double helices. We then melt, anneal the primers, make DNA, and we end up again doubling. So we do this 30 or 40 times, and you've got an exponential amplification that produces an easily measurable and analyzable, very specific DNA. So this is all that PCR is. It allows you to target a very specific part of the DNA. We have entire control over that, and we can make lots of that stuff that is a high fidelity, that is the correct copy of what was present in the chromosome in one copy to begin with. So very powerful and real basis for all of the techniques I'll be talking about. So what are these PCR targets that we would use if we're interested in studying cyanobacterial harmful algal blooms? There are a lot of options, and maybe too many at the moment. And in one of the issues, I think, in applying genetic information to routine monitoring and researching of blooms is to figure out which of these targets and what implementation is the best. But the possibilities are that we can detect essentially all HAB-forming cyanobacteria. We can do that. We can also design those primers in the second line here to target specific genera or specific species, or actually we could even do it for specific subspecies, specific strains. I'll give you an example from our research later. So really here, as I said, we have entire control, and we can deliberately make the PCR rather general, that is all cyanos, or very specific. With that DNA that we've amplified in a test tube, in a very small test tube in a machine called a thermocycler, we would follow that either by DNA sequencing and get the actual sequence of the amplified DNA, or we can do that PCR in a way that's termed quantitative, or sometimes real-time PCR, in order to measure the amount of the starting DNA, the amount of that specifically targeted starting DNA. So basically, we can either do the PCR to get the sequence, or we can do it to measure the amount of a specific DNA. And those targets, there are a number of possible gene targets. The most common ones I've got listed here are the small subunit or 16S ribosomal RNA. This has been used to barcode or identify really all forms of life, and that certainly can be used in cyanobacteria. It's a little bit limited amongst cyanobacteria because they're pretty closely related. The TS is the internal transcribed spacer of the ribosomal RNA, so it's a slightly different part of adjacent to the 16S ribosomal RNA, and that's been very useful. The CPCA targets the cyanin genes. These are pigment genes that are specific to cyanobacteria, and target the spacer between two protein coding genes. The CY genes are the microcystin producing genes that are present in Microcystis and some other genera. At the moment, we don't have many other toxin biosynthetic genes because the sequences aren't known. So, for instance, at the moment, we cannot use this approach to assay the anatoxin biosynthetic genes, but that should be possible in the future. And the possibility is the NIF gene for nitrogen fixation, because some cyanobacteria can fix nitrogen. And so this simply shows what you would get if you do the DNA sequencing. The top colored panel shows a trace where each base is shown by a different colour, and the sequence is simply shown below. Of course, gazing at this doesn't tell you much. There are fairly simple bioinformatic tools that one does need to use, and that takes some training, but it's not at all inaccessible. It's not something that we envisage a like manager would do. This the information gained from DNA analysis would generate these sequences, would be used and interpreted in the form of showing closest relationships and so on that would be useful for decision making. Any questions at this point? Okay, so let's go ahead and use a case study from our own research in the Klamath River, where there are a couple of dams that become hit with predominantly Microcystis blooms that are very toxic during the summer. So here our goal has been to describe that cyanobacterial, mostly Microcystis population, particularly in Kopko Reservoir. That'll be one I'll be talking about mostly. In order to understand what the population looks like and how it changes during the season and between seasons, so that we can develop techniques for the monitoring application of the type that I've discussed, and in order to understand how the bloom develops and why, and see if we can then figure maybe ways to ameliorate the bloom, or at least if we do come across management options, to follow the change in the bloom as that management is applied. So this shows the map of Kopko. It's on the Klamath River that is shown on the top right. That panel there runs from Upper Klamath Lake, which I'll mention a little bit too, in Oregon, across the border, and the reservoirs, the dams, Kopko Reservoir is just south of the border. Taken samples here from near the dam at the CRO1 site, and others have taken splits of these samples and done conventional sort of analyses that are shown, the results of which are shown here. So the solid line represents Microcystis cells per mil. So there's a significant bloom during June in this particular year, and the large bloom in the latter part of August and early part of September. Now, the toxin analyses, the microcystin analyses that were done on the same samples, the amount of toxin. And if you then calculate the amount of toxin per cell and plot it, this gives you the dotted line. Now, this is a logarithmic and exponential scale, as you can see over on the right-hand side. So the large drop that's seen here in the period between August and September is actually about 100-fold drop in the so-called toxin quota. That is the amount of toxin being produced per cell. So either cells are shutting down toxin production late in the season, or there's a replacement of toxigenic cells with non-toxigenic cells. And we'll see that our genetic analyses have been able to provide an answer to those, discriminate between those possibilities. So we can measure the same thing using quantitative PCR, using the so-called Tuckman assay that's listed at the bottom, with PCR that is directed at two genes, the MCYB gene, so that's one of these MCY microcystin biosynthetic genes, a toxin biosynthetic gene, and CPCBA, that's the phycocyanin gene that I mentioned. So this is a single copy gene. It's present in one copy in all bacterial cells and certainly all Microcystis cells. So the ratio of MCYB to the amount of MCYB DNA to the amount of CPC DNA, which is plotted in this bar graph, should represent the proportion of cells that have the toxin gene. It's generally found that in a Microcystis bloom, some cells carry the toxin synthetic genes and some don't. There are a range of strains genotypes of Microcystis that can be present, and one or the other might outgrow its competitor. So what we're plotting here is, as I say, the proportion of cells that carry a toxin gene and should have the capability of making toxin. And we see that in June in Kopko Reservoir, this was very high. This is about 90%. And it starts declining somewhat gradually, but there's a large decline there at the end of August to September, such that in September and October, there's very little MCY DNA around. So there's a different way of viewing the information, or it's the same, give the same information as in this previous slide, which was based on direct toxin measurement. So this type of DNA analysis indicates how you can use the quantitative type PCR to measure the amounts of DNA, and it gives an example of how that can be used to measure the amount of potentially toxic cyanobacterial cells. Now, what I want to go on to then is how you can use genetic means to describe the population. And in order to do this, at the moment it's somewhat labour intensive, so you wouldn't do this for all samples as in routine monitoring, but you certainly would do this as part of a rich program that's associated with trying to understand the bloom and how you might remediate it. And so the PCR is used to target gene sequences as I've described, and then those DNA amplification products are cloned in bacteria to produce what I've got shown in here in yellow, a clone library. The intent then is to separate into individual bacterial cells single DNA molecules derived from Microcystis, or from in this case, or any cyanobacteria, so that we can then grow those bacteria on a plate, spread them on a petri dish, on an agar plate, diluted so that one bacterium can grow into a colony, and each colony is a clone then that carries one PCR product derived from that Microcystis DNA. And then we can pick those, a number of those, and sequence them and generate a library that represents the DNA sequences present in the original sample, represents the population, that genetically represents the population makeup of the sample. In our particular case, we used the ITS, the CPCBA, and the MCY gene to study this population. So we talked about each of those genes before. This is MCYA, which is just the next door neighbour of MCYB. It's one of many genes that are needed for toxin biosynthesis. And generating all of these sequences then allows you to make a tree like this. This is called a phylogenetic tree. It's a bit like the one I showed you earlier. This represents, it's very similar to a genealogical tree that represents relationships amongst our families and so on. And I'll get back to this in a few times, but what I want to point out now is that it roughly shows groupings that are listed here as group 3 at the top left, and group 2 underneath, and 3, 4, and 1. So we can see from this that there's some population structure which is of interest and that I'll go into. This presents the result of analysing the libraries at the ITS target, gene target, and the CPC target on the right. And one of the things we could see is that actually ITS sequences were unique. Nobody had ever found exactly these sequences previously. But in this slide at the top left in that panel, we can see that there's an isolate from Japan and one from the UK and one from Greece that are relatively close in sequence to these Klamath River isolates. So what we have going there in the Klamath seems to be unique in terms of the sequences that have been found so far, but certainly related to worldwide genes. And Microcystis, for instance, as many of the cyanobacteria, are really thought to have worldwide distribution. pretty uniform worldwide distribution. At the TC locus, the situation was a little bit different because some of the sequences had been seen before, and so the black genes are the sequences are the ones that were already in the database from samples from Israel and Spain, for instance, here, and these had exactly the same sequence in that part of the DNA that we analysed as some of the coloured sequences, which were the ones derived from our gene library. So this is the way in which one can disinformation, such as this new bloom found in this lake, which had never before been supported a bloom, is actually very closely related to this bloom that's existed upriver or downriver, and probably represents a spread of those cyanobacteria. So that's the sort of information that I think we'd frequently like to conclude from this analysis. If we go back to the issue I mentioned of the subdivision of the sequences into groups, I've circled at the bottom here a grouping that we've called subgroup 1 of amongst the ITS sequences, a grouping of very closely related sequences. And if we look more closely at how the sequences are related, we see at the very bottom there's just a grey bar. This represents the most commonly found sequence amongst all of our analyses of about 140. There were 140 or 150. There were 4 hits of this exact same sequence. And then if we move up that panel, we see that there are small coloured boxes, each of which represents one base, one nucleotide, one position in the DNA different over about 491. So this is one in about 500 differences that was found in most of the sequences that are dramatically represented in this panel. Towards the top, there are a few that show two nucleotide differences or positions in differences in 3. But you can see in the explanation over on the right, these sequences are, as I just said, very closely related, but they're also relatively infrequent. Now, at the very top is a bar that has many colours. This represents an outlier. This is really no longer part of that group, but you can see that there are quite a lot of differences that in that sequence. So a different way to diagram that is to show the groups as shown here. And actually group 2 should be labelled as group 1. I apologise. What we see in the middle then is that most common sequence that was found many, many times, and very closely related sequences, and quite a number of them, but altogether not as many as the number of times we found the most common sequence. But sequences, this cluster, this group of sequences, this genetically isolated group, quite different from each of the other groups, as represented by the length of the line to group 3 or to group 1, or separates group 3 from group 4. So it's interesting to see that the population A contains a fairly small number of distinct genetic groups, and B, those groups are very comprised of very closely related individuals, so that we should be able to monitor specifically for each group and see how the proportions of the groups change, perhaps during the season. And an example of that here. So using the sequences as I've described and classifying them into the different groups, we saw during the course of a year in Copco Reservoir that genetically, this is analysing at the CPC locus, the CPC, the phycocyanin gene. From June to August, there was very little change in the population of the bloom through the first bloom peak, and the bloom picture was actually quite simple. most of the Microcystis organisms belonged to group A. They were all very closely related. Now, after into August and later, we can see that there was a massive replacement of group A, which all but disappeared, and the appearance of a number of different genetic groups of different strains. So we see here that there was a strain turnover, and that this actually corresponds with the transition that I described earlier from toxic bloom to much less toxic bloom. So probably group A represents toxic organisms, and the other groups, such as B1 and B2, non-toxic. The situation is similar, but a little bit different in the details if we look at the ITS gene. But then we see that in June, the population is relatively simple. In this case, in October, the population looks relatively simple, very simple at this locus, but that there's a turnover in between. So the point, I think, here is that we can use genetic means to understand how the population is changing, and it changes in a very important way, transitioning from being very toxic to being much less toxic. That's certainly something that we would like to manage if possible, because it's certainly better to have a non-toxic bloom than a toxic bloom if there's any bloom present at all. And so by understanding what the subpopulation structure of a bloom is, we certainly have the possibilities to perhaps suggest a management means and to track management changes as we implement them. So shown here, where we've gone back to the phylogenetic trees, is the sequences distributed according to their closest relationships, but coded according to time. And see very graphically here that the early season samples all represented by which are represented by warm colours, are clustered at the tops of these two phylogenetic trees at the two different PR loci, where late season sequences are clustered at the bottom. So it very graphically illustrates that we've had a big shift in the population, something that you would never have been able to predict from microscopic observation. Furthermore, some of the sequences present in these trees are coloured with purple. You should be able to pick those out. These are taken from Upper Klamath Lake. That's the lake close to the headwaters of the Klamath River up in Oregon, whereas all of the other sequences with the different colours I mentioned just a couple of minutes ago are from the Kopko Reservoir downstream. Currently, there is a limited, very limited problem with Microcystis growing in Upper Klamath Lake. But it was actually quite a concern to find through these analyses that the Microcystis strains that are present in Upper Klamath are similar, and some are probably identical to the ones growing in Kopko. So that if the Microcystis Microcystis population in Upper Klamath were to expand, currently it's present in much smaller amounts than Aphanizomenon. Mostly there's a non-toxic Aphanizomenon bloom. But if the ratio were to change, the bloom were to become predominantly Microcystis, then quite likely it could be very toxic, and this would be a real problem. Okay, we're just about the end. So my contention is that the sort of approaches that I've described here can be very powerful, and that really it's time to start to work to implementing the wonderful academic knowledge that's known in the realm of cyanobacterial genetics to applied routine monitoring, or at least understanding blooms. And to get there, we do need to build genetic databases. And this reiterates, I think, that all the points that I think I've already mentioned, that are the advantages that I anticipate from genetic analysis of cyanobacterial blooms. So I think I'll stop there and see if there are any questions.